Dear students, welcome to my video tutorial Brush Up Zoology, and we are taking the topic Immunology Basics. In my last lecture, we talked about cellular barriers, and amongst the cellular barriers, we talked about phagocytic barriers. We also talked about B cells, T cells, NK cells, eosinophils, and now we are going to talk about auxiliary cells, which play a role in inflammation. And whenever inflammation happens, certain infection is there, the body tissue responds and inflammation starts. And leukocytes are attracted towards the site of inflammation. In addition to that, there are certain cells such as basophils and the mast cells, as well as the platelets. These also promote inflammation. Basophil and mast cells both have granules, which are these granules, they release certain mediators, and mediators, they promote inflammation by releasing histamine in the surrounding tissue. Similar function is done by the mast cell. Mast cells are present adjoining the blood vessels, whereas basophils are the mobile cells, they circulate in the blood. And how they act, both these, whenever histamine is released by these cells, histamine binds to the receptor, or any such mediator would bind to receptor on the nearby capillary or a blood vessel. And similar, there is another kind, kinins. The function is almost same, and that dilates the blood vessels and increases the permeability of the blood vessels, by which the fluid, it leaves the blood vessels, and in the localized area edema is formed, swelling of the particular area, and that also in one of the inflammatory reaction that happens, and that further dilutes the toxins released by these foreign particles or any kind of pathogen. The similar kind of the similar type of the mediators, which are kinins. And kinins, what are kinins? These are small peptides, and these are in inactive form. They present in inactive form in blood plasma, and whenever there is tissue injury, they are activated. They change into activated form, and then they would result in, again, they would act in the similar fashion as the histamine does. They would result in vasodilation. One example of kinin is bradykinin. The bradykinin, it stimulates the pain receptors in the skin. So that is also a kind of immune response of the body. Then next is the soluble mediators. Mediators of immune system, or they are also called as the chemical mediators of immune system. The cellular mediators, they are antibodies and cytokines, which are produced by lymphocytes. There can be other molecules which are normally present in the serum. They are present in the serum other than antibodies and cytokines. And there are certain proteins present in the serum. Proteins are present in the serum whose number increases rapidly during the infections. Certain infection, these number would increase. And such proteins whose number increases during infection, these are called as acute phase proteins. So this is another type of chemical mediator which is found in the protein, which are found in the body within the serum. For example, one example of such proteins is CRP, C-reactive proteins. These are C-reactive proteins. These are called so because they are capable of binding to C polysaccharide, which is a on cell wall of some bacteria and fungi. So this protein, its function is the CRP or cell reactive protein. It helps in picking up the bacteria, or let's say certain foreign particles, and then helps in phagocytizing them by activating the complement system, which we would be studying now. So that is the role of CRP. CRP activates the complement system, and then it helps in phagocytizing of the, let's say, bacteria or any kind of pathogen. So this is the role of acute phase protein. One example was CRP. And whole this process is called as opsonization. And such molecules which promote opsonization called as opsonins. Example is acute phase protein. One example CRP, or another example for opsonin, let's say antibodies, they also play such a role. So next is the complement system. What is complement system? Complement system, this system participates in both innate immunity as well as in adaptive immunity. The system is a group of at least 20 serum proteins. And these serum proteins, their function is to control the inflammation. These different components of such a complement system, they interact with each other and with other elements of the immune system, and there are basically two type of complement system. One is the alternate pathway, or it is also called as alternative or bypass pathway, and second is the classical pathway. This particular pathway operates in innate system, innate immunity, and this system is activated in, how it is activated? This system is activated by, let's say, endotoxins, by microbial cell wall, components of microbial cell wall, or other such components of microorganisms. And once it gets activated, the system gets activated, the microorganism, suppose this is certain microorganism, it gets coated by these complement components of complement system, and ultimately it would be phagocytized by a phagocyte. And we say that this microorganism gets opsonized. So process is opsonization. So this is how the alternate pathway it operates. Whereas the second type of system, that is the classical pathway. The classical pathway, it is activated by antibodies or also activated by mannose binding lectin bound to pathogen surface. So it would be activated by these two components. Now how the complements activation system works. The complement system, basically its activation is basically a cascade reaction, chain reaction. Where one component, let's say certain component C1, it acts as an enzyme on the next compound, and then certain enzyme is generated, and which would help in activating third component, and so on and so forth. So it is a kind of chain reaction where one component would act as enzyme for activation of another in a sequence, and in sequential manner the reaction is set up and whole system is activated. Something like the cascade of blood clotting system. So how now the complement system it works? Complement system, activation of now once the complement system it has been activated, so activation of complement system, it generates protein molecules or peptide fragments which have following effects. What effects it has? Opsonization, you will see here, this is a complement, it has been activated and it would have different effects. When I said opsonization, how? Suppose this is the bacterium, gets coated and it gets attracted by the phagocytes get attracted and it is phagocytized. So this is opsonization. Then is chemotaxis. That means attraction of phagocytes to the site of infection. These phagocytes are attracted to the site of infection. So that is chemotaxis. And third is lysis. That means the damage of plasma membrane of all the cells, let's say the bacterial cell or the enveloped viruses or other organisms, these would be lysed and they would ultimately, that is called as the lysis. So these are main three mechanisms by which the complement system it operates or its mechanism is like that. Where in addition to that, another effect that takes place, that is the increased blood flow to the site of activation. Blood flow to the site of activation due to increased permeability of capillaries to the plasma molecules, and then thence the that would lead to. And fourth is the next is fifth step is the release of inflammatory mediators. So inflammation also starts in this is also a part of inflammation, and that is also a part of inflammatory reaction. So these mediators like histamine, kinins, etc., these would be released. So this is the working of complement system, mechanism of complement system. So that is all for now.